We have searched the Paramecium database for sequences indicating the presence of calcium release channels type inositol trisphosphate receptors and ryanodine receptors. We concentrated on prominent domains such as an IP3 binding domain, a ryanodine IP3 receptor homology domain, RIH, the pore region and its selectivity filter between the last two transmembrane domains. We found a total of 34 candidates grouped in 6 subfamilies, Roman 1 to 6. Only type 1 to 3 contain a clear-cut IP3 binding domain, and only some contain a RIH domain. From the dense ramifications in the cladogram on the right side, it becomes evident that diversifications within the subfamilies are largely due to recent whole genome duplications, and the respective paralogs can serve gene amplification. Only type 2 and 4 subfamily members have been characterized in more detail as IP3 receptors and as ryanodine receptor-like proteins. The next slide presents the region between the last two transmembrane domains. Besides an occasional expansion, there are evident identities and similarities. The selectivity filter mostly contains the amino acid motif GXGD, X being mostly isoleucine in both receptor types, whereas normally GD is characteristic of IP3 receptors in higher eukaryotes. The next slide presents the intracellular distribution of the different calcium release channels, Roman 1 to 6, standing again for the respective subfamilies and Arabic numbers for the subfamily paralog actually tested. Note that the different calcium release channels are associated with specific organelles participating in intracellular trafficking, where local calcium release can trigger membrane interactions. Some organelles contain more than one channel type. Examples of immunofluorescence are presented on top. The ryanodine receptor type channel is localized to the alveolar sacs, established cortical calcium stores activated during exocytosis stimulation. A true IP3 receptor with binding capacity for radioactive IP3 is associated with the contractile vacuole complex. Here it is constitutively active and serves fine-tuning of calcium sequestered into the organelle by reflux into the cytosol. The next slide presents similar sequences in other organisms, mostly only with fragmentary similarities. These are most pronounced in the C-terminal region with the potential pore domain. However, no analyses were conducted in similar detail with other protozoa as yet. The final slide summarizes our most essential findings, stressing features characteristic of IP3 receptors and ryanodine receptor-like proteins in Paramecium. Beyond that, these cells also contain additional calcium release channels not addressed here.